Auzu billahi minash shaitanir rajim. Bismillahirrahmanirrahim. So, moving into the last week of our recorded online teaching session, today we are going to start about. It is not explicitly separated in your course contents given by the department, but I have divided the course into different sections. You could always say this part is not from history. We have already discussed many things that were from industrial biotechnology, but today we are going to talk about fermentation, what are fermenters and what are bioreactors, a bit about ethanol production, a bit about antibiotic production, and a bit about downstream processing. And you will see all of these are interconnected because they all belong to more or less the same phenomena of industrial biotechnology, that is for the production of products, synthesis of products, therapeutic or non-therapeutic. And this is the image of, you can see here, this is Penicillium chrysogenum, which is used for the synthesis of antibiotic penicillin. This is in transmission electron. This is also in transmission electron microscopy. Both of them are SEM images. So Penicillium chrysogenum, and this is the budding yeast. You could see the buds on the surfaces of Saccharomyces cerevisiae. Okay, so industrial biotechnology. What areas of industrial biotechnology are covered? Medicine has a very important role and a connection with industrial biotechnology. For example, synthesis or the biosynthesis of antibiotics is through industrial biotechnology, and it has all its applications in medicine. Also, some applications in livestock, but that could also be included in medicine. Then the development of new vaccines is all industrial biotechnology. After the clinical trial, the clinical discoveries, all the molecular biology and the biochemistry techniques in the lab would eventually have to come to industrial biotechnology to produce millions and billions of shots of vaccines for detection, for the prevention towards many diseases. One case which is on the lips of everybody these days is the synthesis of vaccine for COVID-19, the SARS coronavirus 2. And after these trials, as far as I remember, 5 of vaccines are already in human trial, either 1 or 2 or 3. And either of them is approved, we will have to go to industrial biotechnology to have them synthesized on commercially large scale so that the whole world can benefit from it. Then development of new processes for higher production so that we have efficient production, loss of energy and loss of resources are minimized, more products. Food processing is very important so that we can preserve the food, we can pack them in clean atmosphere so that their shelf life could be increased. This is also in industrial biotechnology. Then they have a role in beverage industry because a number of beverages are synthesized or produced through industrial biotechnology. And then industrial biotechnology could be applied to improve food quality. Now, what is respiration? Before understanding fermentation, it is very important to understand what is respiration. As a kid, when you went to primary school, we were told, or you were told, that the taking in of oxygen and expelling out of carbon dioxide is respiration. That's true. But in a biochemical sense, the breakdown of glucose molecule in the presence of oxygen is respiration. Now, if we complicate things and go and dig a little more or a little deeper, we would find out that respiration can also be of 2 types: aerobic respiration and anaerobic respiration. Now, as the name indicates, aerobic respiration happens only in the presence of oxygen, and anaerobic respiration should be without oxygen. A respiration always starts with a process of glycolysis, which is actually anaerobic part. And after that, if it's going to be respiration. It has to go and start taking place through the triacetic acid cycle, also known as Krebs cycle in mitochondria. Or if it has to go anaerobic way, then it has to go and it will be fermentation. And aerobic respiration through electron transport chain, through glycolysis, where we have the substrate level phosphorylation, and also in the Krebs cycle substrate level phosphorylation, while in the electron transport chain we have oxidative phosphorylation. We yield a lot of ATP molecules. On average, for one glucose molecule we have 36 or 38 ATP molecules. Theoretically, the number is controversial. Some books and some scientists are of the opinion that it's 36, while others are of the opinion it's 38. So to be on the safe side, we say 36 to 38. While in case of anaerobic respiration or fermentation, it's way less. It's 8 to 12 ATP molecules, because here we don't have any oxygen involvement. Okay, so fermentation technology. Now, there are two terms. One is fermentation, and the other one is fermentation technology. So they are a little different, yet the same. Fermentation technology is not a new technology. It's one of the oldest biotechnological processes. And literally, it is derived from Latin verb fervere, which means to boil, and the appearance of fruit extracts or malted grain, which is because of the action of yeast during the production of alcohol. This is called fermentation. Fermentation has different processes, but normally it is a process, a biochemical process, where there are a number of changes caused by either the organisms or their products. When we talk about their products, necessarily in this case for fermentation, it means enzymes. And there's effervescence and heat generation. Now, if we define fermentation in terms of microbiologists' point of view, they would say any process for the production of a product by means of mass culture of microorganisms. So they would involve microorganisms in everything. Now, if we talk to biochemists, they would say it's an energy dependent or energy generating process where organic compounds, both they act as electron donors as well as acceptors. Fair enough, that's true. But how can we generally define the term fermentation? It's an anaerobic process where we have the production of energy without the participation of oxygen or any other inorganic electron. In biotechnology, microbiological concept is widely used because whenever we put bacteria or fungi or some other prokaryotes for the production, for the synthesis of important compounds, therapeutic proteins, or related biomolecules, we simply call the process as fermentation. So any industrial biotechnology product process is called fermentation. When we say fermentation in terms of biotechnology or industrial biotechnology, it does not necessarily mean this fermentation that is connected to respiration. In some cases, there will be involvement of oxygen, but we will still call it a fermentation. And we will still be doing this whole process in a chamber that we would call fermenter. But does not necessarily mean that there will be no oxygen. In general, fermentations can be divided into 4 types. The production of biomass, for example, baker yeast, proteins, lactobacillus, production of extracellular metabolites, chemical and compounds. This includes secondary metabolites, intracellular, all these genetic engineering through which we do produce, for example, insulin, for example, the growth factor 9, for example, the antiviral proteins, which we call interferons, and the transformation of a substrate in which the transformed substrate itself is the product. For example, production of ethanol, in which we use corn, and then we produce ethanol. Okay, now this is a chart that represents the essence of fermentation. The respiration process always starts with the C6 molecule glucose, and when glucose molecule, it goes through glycolysis. There's a small amount of energy produced through substrate level phosphorylation, because there is a net of 4 ATPs would be gained, and then you convert it to pyruvate. After pyruvate, this pyruvate has to be either converted to lactic acid or ethanol. These are two different types of fermentation. Now here we are excluding the possibilities of aerobic respiration, and we are not connecting this to the tricarboxylic acid cycle in the mitochondria. In animals, in humans, we have lactic acid production in some bacteria and fungi as well. In plants and in many bacteria, most of the bacteria and fungi, they would go for the synthesis of 2-carbon ethanol production. When we do a lot of exercise, running or gym, we feel fatigued. That fatigue is because your body needs a lot of oxygen, and because a lot of hard work is being carried on, and you cannot take in sufficient amount of oxygen, through which, because of which your body shifts to fermentation and starts generating rapidly ATP molecules, where you have fewer number of ATP molecules, but the process is fast enough through a process where lactic acid is produced. And the lactic acid production and synthesis, it accumulates in your muscles, due to which you feel fatigued. And this is why when you take rest, slowly and surely the lactic acid is broken down again, and you, after a few hours, you feel okay and you don't feel fatigued.